Hello, class. Welcome back to Biology 1110. We are in Chapter 5. We are finishing up Chapter 5, talking about our biomolecules. And the last one we have are nucleic acids. Nucleic acids are molecules that store, transmit, and help express hereditary information. So hereditary information, that's the stuff that you inherited from your parents. So we have all these proteins we just talked about, and the proteins are going around doing all of those jobs that we talked about. So pretty much what is keeping your body running are proteins. But behind the scenes, what is keeping those proteins being made and being created and keeping the information for that are our nucleic acids. So the amino acid sequence of a polypeptide, which is what your proteins are made of, is programmed by a unit of inheritance called a gene. Your genes have all of the instructions to make all of your proteins. Genes are made of DNA. DNA is one of the nucleic acids we're going to talk about. And the monomers of nucleic acids are nucleotides. When we're talking about DNA and when we're talking about RNA, the monomers of nucleic acids are nucleotides. So there's two types of nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid, so D for deoxyribo, N for nucleic, A for acid, and RNA for ribonucleic acid, R for ribo, N for nucleic, and A for acid. So they're acids, they are made of nucleotides, and they're going to have a sugar, either deoxyribose or ribose. DNA has directions for its own replication. We're going to talk about how it is double-stranded, and those two strands can separate and be copied to become double-stranded again. And then DNA will also control the creation of RNA. So DNA can be read in order to make a copy of mRNA. And then reading mRNA will give you the ability to create proteins. So mRNA controls protein synthesis. This whole system of DNA being read to make RNA and RNA being read to make proteins is called gene expression. And it always goes in that order. DNA is used as a template for RNA and RNA is used as a template for protein. Always that order. Each gene along a piece of DNA will direct the synthesis of one specific messenger RNA. And the messenger RNA molecule will interact with all of the stuff going on in the cell to direct the production of a polypeptide. So here in the picture, my pointer's not working. You can see up the DNA here, this little blue ribbon up at the top left of the picture, is going to direct the synthesis of RNA. That RNA is going to leave the nucleus through a little nuclear pore and move into the cytoplasm. The RNA will interact with a ribosome, and ribosomes are going to build a protein. So as the RNA is moving through the ribosome is putting together this polypeptide. It's grabbing these little amino acids and putting them together into a polypeptide. So the flow of information is DNA up at the top, RNA in the middle, protein here at the bottom of the picture. DNA, RNA, protein. Nucleic acids are polymers called polynucleotides, and each polynucleotide is made of a monomer called nucleotides. So each bead on that string is a nucleotide, and the whole pearl necklace is a polynucleotide. Or each little Lego block is a nucleotide, and the whole block tower is a polynucleotide. Each individual nucleotide is made of 3 pieces. In the center image here, there are 3 pieces. You have a nitrogenous base, that is the orange part of this middle picture, a sugar, a pentose sugar, so pentose tells you it has 5 carbons in it, a pentose sugar, and one or more phosphate groups, so that's highlighted in yellow. Every single nucleotide has those. And if you have that nucleotide and you lose that phosphate group, then what you have left is a nucleoside. So the sugar with the base attached would be a nucleoside. Once you add the phosphate group, you have a nucleotide. So nucleoside is a nitrogenous base plus a sugar. And there are two families of bases if we're looking at just the bases. There are pyrimidines and purines. So you will need to keep these straight. Pyrimidines, I remember as being the longer word, but the smaller shape. And purines are the shorter word, but the bigger shape. So pyrimidines, that's what's at the top of this picture. We have cytosine, thymine, and uracil. Cytosine, abbreviated as C. Thymine, abbreviated as T, only found in DNA. And uracil, abbreviated as U, which is only found in RNA. Those are our three pyrimidines: cytosine, thymine, and uracil. And they are made by one central six-membered ring. One little, that orangey brown hexagon is that six-membered ring with various little functional groups coming off. Purines are going to be adenine and guanine. They're at the bottom picture here. Adenine and guanine. Adenine is abbreviated A, guanine abbreviated G. And they are made of a six-membered ring fused to a five-membered ring. So you have the hexagon fused to a pentose. And then when you put the base with a sugar and a phosphate group, then you have the nucleotide.
DNA and RNA are nucleic acids. It's called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose.
Okay, that's one important distinction that you need to remember. This, as we're talking about differences between DNA and RNA, you need to remember that they just showed you there's a difference in the sugars that they have.
Has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil.
Okay, so there's another difference between DNA and RNA that you need to remember.
The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and help.
There's another difference between DNA and RNA. Make sure you're paying attention to that.
Held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, so if you want to go back and watch that video in its entirety without me pausing for commentary, just go back. You can download the lecture video and play it. It won't play within D2L, but if you download the lecture video, you can play the videos.
DNA and RNA are nucleic acids.
Sorry, going the wrong way. Okay, so DNA has a pentose sugar and RNA has a pentose sugar. Pentose, means 5, and the "-ose ending tells you it's a sugar. So it's a sugar with 5 carbons. So you can see them numbered in both pictures. DNA is called deoxyribose because that oxygen has been taken off. It's been deoxygenated, so that it is deoxyribose. And in a nucleotide, when you are making a nucleotide, that number 1 carbon, so see where it says 1 and has a little dash, that's the 1' carbon, and that's where the base is going to be attached. And then see over here where it has a 5 and a little dash, that's a 5' carbon. That's where the phosphate is going to be attached. Nucleotides will be joined together to build a polynucleotide, to build either a long RNA or an even longer DNA molecule. Those nucleotides are going to be bonded by a phosphodiester linkage. That's a big word. You don't have to know what it means, but that is how nucleotides are bonded together. And that bond will be a phosphate group linked to the sugars of the 2 nucleotides. And that will form this sugar phosphate backbone. So if you went along the backbone, you'd see sugar phosphate, sugar phosphate, sugar phosphate. And then you'd have the bases sticking off of that. So like in this picture, you can see the blue ribbon. That blue ribbon stands for sugars and phosphates. And then these orange bases sticking out, the A, T, C, and G. The sequence of those bases is unique for every gene. Just like the sequence of letters is unique for every word, and that's what makes it a word, the sequence of bases in every gene is unique, and that's what makes it that specific gene. DNA molecules have 2 polynucleotides, and they give you this pretty little curl, kind of like a rounded staircase. What are those called? Circular staircase. So these 2 strands that are spiraling around this axis, forming a double helix. But they're running in opposite directions. So if you imagine one line is facing forward and one line is facing backwards. One line's going up, one line's going down. Because even though they're made of the same things, they're going backwards from each other. We call them antiparallel. So the 2 strands are antiparallel to each other. And one DNA molecule will have many genes. So we have 23 pairs of chromosomes, but each of those chromosomes has many, many genes to give us all of the proteins that we need in our body. On the bases in this DNA, they're going to hydrogen bond with each other to keep those 2 strands together, connected. And even though those individual hydrogen bonds are weak, between all of the bases together, that forms a nice, strong bond between the 2 strands, so the 2 strands of DNA stay together. But they're going to hydrogen bond in specific ways. There's always going to be adenine bonding to thymine, and it's going to be cytosine bonding to guanine. So the little mnemonic I learned is apples in the tree, so apples in the tree, A and T, adenine and thymine. Cars in the garage, cars and garage is CG for cytosine and guanine. That's called complementary base pairing. So this is going to be what makes it possible for DNA to be split apart and copy itself, because if you split apart the strand and you see an A on one strand, then you know when you're building the next strand it needs to have a T there. And if you see a C on one strand, then you know it needs to be paired up with a G. So the cell is able to copy all of your DNA very accurately based on this consistency in base pairing, this complementary base pairing. When we compare RNA to DNA, RNA is not RNA is single-stranded. It's not going to form that pretty spiral staircase that DNA made. There can be complementary base pairing of RNA molecules to help it fold up into its shape, or between different RNA molecules. In RNA, we take the thymine out and replace it with uracil. So wherever there was a thymine in the DNA, the RNA that's copied off of it is going to have a uracil instead. And then another difference is DNA is always the shape of a helix, but RNA molecules are going to have some various different shapes that they take.
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the 2 sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
Okay, so that is the end of this chapter. So please make sure you are comfortable with everything we've talked about in chapter 1, which is kind of the foundations of science and the scientific method. Chapter 2 was atoms, structure of atoms, and how they react. Chapter 3, we started talking about water and the different properties of water. And in chapter 4, we talked about carbon and how carbon is a backbone, different functional groups. And in chapter 5 here, we've talked about all of our major classes of macromolecules. Please make sure you're very comfortable with all of those because we will be having your first exam over these chapters. There is a review packet. It's like 7 or 8 pages long, has all the questions you need to be able to very comfortably be very comfortable and familiar with. So you can be filling that out. We will talk about it in class and any other questions you have. So either make sure you bring those questions to class or send me an email if you're not able to make it to class. The exam will be in class, so make sure that you do have time to come to class for your exam. And I will see you later. Bye.